like the flex questions. I really like them, but others don't. So, so you know, the A and B or C and D or none of the above, you guys seem to really dislike those kind of questions. So I've tried to make them to actually even remove some of the none of the above as well, because when it's none of the above, the three, typically the three, you know, the A, B, and C above, you have that can be tricky. So everything has to be correct if it's all of the above. So that's a little bit more fair. There's actually a science, an art and a science to writing these questions this way. So
We can start. Since I'm recording, it's 11:05. I've opened up screen share. If you'd like to share your screen, Dr. Hogan.
sure, sure. Let me go close my office door real quick.
Okay.
All right. I found if I don't do that, I have interruptions that could be embarrassing. So let's not do that, especially not with it being recorded. So let me.
It is being
Um,
recorded.
yes, that I don't want embarrassing things recorded. So all right, let me. You guys know I'm technologically inept.
No, you're not.
So all right. So we can start here. Let me see if that still works. Does that look right to you guys?
Yeah, it looks good.
Okay. So the first thing I always tell people every year is, even though it's a 1 hour course, please study for the exams. Please do not, think, oh, it's a 1 hour course, I don't have to study, I'll just go in. Please don't do that for your own benefit. Okay. Because you'll end up with answers like this: math test, Bob has 36 candy bars each, 29. What does he have now? Diabetes. Bob has diabetes. Those will not get you points on the on the exam. I think that kind of goes without saying. Um, Best solution for overpopulation: the Hunger Games, hard and soft water, ice. These are not obvious. These are on the internet. What ended in 1896, 1895. So, I haven't gotten these. I've gotten some that have been close, and we don't want any of those because they're not going to help you with the grade. So, example questions. So, filaments of actin elongate A, B, C, or D in a symmetrical manner? No, it's not symmetrical. By hydrolysis of peptide bonds. Huh. Uh, no, that's not a peptide bond either. By hydrolysis of GTP molecules. Now, you could be fooled by this one because it does utilize, this process utilizes energy, right? But it's not GTP, it's ATP. What uses GTP? Well, microtubules use GTP. So it's clearly none of the above. Okay. So, this is like I said, I don't have many of the none of the above questions, but hopefully this will kind of scare you guys into studying a little bit. Okay. Um, Now we'll go to next question. So again, I'm not doing this to be tricky, and I get this in some of the kind of student comments every year at the end of the course. Well, the questions were really tricky. Well, there can only be one correct answer, right, for a multiple choice question, which means by default, I have to put, answers in there that are incorrect. It's not me being tricky. And again, I do look at the metrics that are recorded for what percentage get these questions correct and which ones don't, the percentage that doesn't. And it ranges from between 60 and 80%. Now, if it's on the low end, I will tweak the question based on, okay, 50% of the class got this question correct and, 50% did not. Well, I see that as I haven't done a good job with formulating the question. Okay. it's too tricky or there's some wording in there that trips people up. So I do try and refine them. And that's actually why ExamSoft is great for us, because we can refine these and we want to shoot for about 70 to 80% of the class actually getting the correct answer for it. So let's look at carbohydrates. So they are not the building blocks of proteins. That would be an amino acid. Okay, so take these individually and look at them and think about them. Okay.
Ashley, can you mute, please?
All right. So amino acids for A. So you know that's not the answer. B, components of nucleotides. Okay. Okay, that looks good. Now let's look at C, form mammalian cell walls. Now, does anyone know of a mammal that has cells that have cell walls? I don't think so. So clearly it can't be A and it can't be C. So let's look at D, store electrical energy. You might look at that and that might trip you up, store electrical energy. But we didn't talk about anything in my section that really dealt with electrical energy, okay? What we talked about was chemical energy. So when you eat a Snickers bar, like Bob eating candy bars, I can't charge my phone after I eat, right, a chocolate bar. I can't stick my finger in the charge outlet and charge it up. So clearly that's chemical energy. All right. So the answer is components of nucleotides. So if you go through the early lectures, you'll have this information, right? You'll see it. And in fact, if you remember, I repeated some slides in multiple lectures. And I said at the time, if you see a slide in more than one lecture, you're probably 90% likely that you're going to see something about that on the exam. Okay. So when you go through and study, if you notice a slide repeated, make a note of it. Pay attention to the slide. Okay. I also got some slides where I boil it down to the essentials. Okay. So let's look at another one. Exons of genes may contain. So before you even look at the A, B, C, and D, what is the exon of a gene? So again, I'm walking you guys through how I design the questions, so it will hopefully help you pick the correct answer a little bit easier. So what is the exon of a gene? So that is the coding region of a gene, right? Well, if you've gone through the genes lecture, which is a very important lecture to focus on, it's not the only one I want you to focus on, but you definitely need to go through that lecture again because it will be a major focus on the exam. If that's a coding region, you know it's not an enhancer, right? And you know it's not a promoter, okay? And you know it's not a polyadenylation signal sequence because it encodes for the actual, that is the amino acid, does result in the amino acid sequence of the protein. So therefore, it's got to be none of the above, okay? Then let's look at a true-false question. And this is the last one I got. Enhancers are sequences present on messenger RNAs, excuse me, that aid in initiation of translation, okay? Is that true or false? Well, let's look at the question again. An enhancer is a sequence present in or on messenger RNAs. And when you go back and you read through that lecture, you will notice that an enhancer is not a sequence that's in the messenger RNA. What is a messenger RNA? That is the basic component that encodes for or allows a ribosome to create a protein with a certain sequence, right? That's the final instruction set, if you will. So you know that you're not going to have anything in there like an enhancer or a promoter or anything like that in a messenger RNA. So by default, this is false, okay? So does that make sense to everyone? You guys kind of see how it's always better when we're in person because I can see people nodding, or I can kind of see, the shock and awe look on people's faces when they see this. Um, so you guys feel free, ask me, any questions you've got. Um, I'm here to help answer them if I can.
Um, Dr. Hogan, I know we talked about it briefly in the techniques lecture, and then it was kind of mentioned even in Dr. Gogol's lectures later on. Could we go over just like what flow cytometry is? I don't know if I fully understand that.
Sure, sure. So, and I'm glad that you asked that question, and this is why. So the tools and techniques lecture, the questions that I may ask you about those in that particular lecture, those tools and techniques, are not going to be very drilled down questions. I've tried to make them more kind of applicable. So if you don't quite understand the technology, fully, but what you could use that technology or that tool for, that is really what I'm looking at. So and the reason I do that is, in other words, if you all are wanting to have a test done on a patient, is to say it's a cat, for example, and you want to see what the CD4 T cell, and it has FIV. You want to see what the CD4 T cell count is. Is it up? Is it down? Are there basically no CD4 positive T cells? In order to get some prognosis of the case, what you would want to use flow cytometry. So that was one of the examples that I use in that slide set, right? So to kind of get to flow cytometry real quick. So what that is, that's basically, think of it as a machine that lines cells up in a single file manner. Those cells pass through basically a flow cell with a laser that shoots right through it. So every time one cell goes through, it passes through the laser beam. And if it's labeled with something, that's going to be caught by the electronics in the machine. It's called a photomultiplier tube. That digital signal is then going to be counted and put on the computer screen where you can see it. Okay. So that's how you're able to determine what types of cells are in your sample, right? With the labeling that you do, monoclonal antibodies or other labels. And you can get a finite number, right? So out of 1 million cells, what percentage are CD4 positive? Well, if it's 0, in the case that I outlined, the cat with FIV, the prognosis is very, very poor, right? There are basically no CD4 T cells left as a result of the viral infection. Okay. Now, if that counts good, close to normal, then the prognosis is quite good, at least for the time being, right? So that's sort of flow cytometry. And again, it kind of helps you guys when you go back to study, at least for that particular lecture, kind of what I'm looking for is you have these tools, what can you use them for, right? So at least you have an idea if you need to do a test, how does that test kind of work? It doesn't mean you have to know how to go in the lab and do it like me or Bob. It just means that you have an understanding, a basic understanding of it. Does that help? Awesome. Okay.
Can I add just one thing about the flow cytometer? Because it's something I also emphasized in lecture, and you're going to see it again in the spring when we talk about immunology. But Dr. Hogan did a great job describing how the operations work. There are multiple PMTs, we call them photomultiplier tubes. One of the first discriminations that machine does when the cell passes through is it assesses two things. It assesses the size of the cell and it assesses the surface area. We call it granularity. So and then what it does, it allows us to discriminate the populations of cells. So if you can reflect on what you learned in histology so far, And cytology, you know that cells have different sizes and they have different shapes, morphologies, and granularities. So with what Dr. Hogan's saying is it allows us not only to look at the fluorescence, which he's talking about, but we can discriminate different populations of leukocytes based on their size and based on their smoothness or roundness or granularity. And we toe that into when we look at cytology. So they complement each other. So if you're in clinical pathology as a clinical pathologist, just to extend that further to cancer, which is something obviously we're also interested in, is it allows you to discriminate those different populations. If it's a lymphoma, like a T cell or B cell lymphoma, which some of you wrote about, or just a different type of leiomyosarcoma or hemangiosarcoma, they can then look at those cells as they do using flow cytometry to characterize the immune response, but also look at the phenotypes, the protein receptors that they're looking at, or they're trying to ask the question, do these cells upregulate the expression of VEGF or something? So it's a very versatile tool. I want to emphasize that. So what he's giving you in respect of FIB is one, but we use it in so many different facets of diagnostics, which is really a very powerful tool. So if you're interested in that direction, so it gives you a little bit more perspective. Any more questions for Dr. Hogan? Guys are quiet.
Yeah, I noticed how many cameras are on and how many aren't.
Well, part of Jeff, is their Wi-Fi connections aren't always the greatest, so it really taxes their Wi-Fi. So shutting off the video and just listening gives them the ability to stay connected.
Ah, gotcha.
Yeah.
Gotcha.
Someone else was saying something.
Yeah.
Yeah, I had one question about mitotic index. You mentioned it and then said you wouldn't talk about it because we had talked about it in histology, but we didn't talk about it in histology. So just how in depth do we need to know the mitotic index?
You talking to me? I mentioned it, right?
Yeah, I forget exactly which lecture it was
I think it was probably me and mitosis. You just need to know generally what it's used for. That's basically it. And you'll get it in ClinPath, but it's and actually if you have a chance to look at some of the, I mean, you don't need them for the test, but I finished all your reports, as you already know, and I sent all the grades out. I uploaded all the reports into ELC, and it's there in a file at the very end of the of our course. So I would strongly encourage you to download those in the near future so you have them on file. As I said, there's 43 of them. Some of them do mention about mitotic figures. So essentially what clinical pathologists do when they're staging cancer and looking at efficacy similar to FIB is they'll do cytology and they'll look at the number of cells that are undergoing mitosis, and they'll relate that based on the number of cells that they see in that field of vision relative to that cancer. And they come up with a mitotic figure, a number. And I gave that to you on the slide. And that's kind of what I want you to know, general specifics. They'll give you a lot more information, or they should, when you get into ClinPath, which will be coming in next year, okay? But by understanding what mitotic figures, mitotic index is and what we use it for, I mean, that's really the gist of what I wanted you to appreciate. And it's pretty well spelled out in that slide.
Okay, thank you.
Yeah, no problem, Sarah. All right, is everyone ready for my part? Okay, I'm going to share my screen. And here we go. I'm going to throw this over there. All right, so I tend to like fill in the blanks because I'm not 100% behind multiple choice because I don't think life is multiple choice. You know that story. I've mentioned it before. So we're going to go through some basic questions. I've actually taken some of them, fill in the blanks out. I converted a couple of those to multiple choice. So just to give you a perspective of how many questions we have, as Dr. Hogan indicated, his questions are multiple choice and true false. I have multiple choice. I have matching, and then I have fill in the blank. And I have one actually case question, which I call my mind bender question, which I have given you an opportunity to write your answers. And I would encourage, and we'll talk about that when I'm finished going through these with you all. Because I think it's important, you know, as future clinicians, you need to have recall. You need to also be willing to take your knowledge and actually apply it. So I've created an interesting case that's focused around cancer, so I make it relevant. And you've all actually contributed to your report in that. So you should be very relatively comfortable. And I've given you an opportunity to really think. And in some respects, I'll give you a little bit of a hint. There's no 100% one right answer, but if you defend it well and you explain it well, you'll get credit. So I want you to kind of step away from it as a test, but start thinking about if you were in the clinics and you were being presented with this case and these opportunities that are presented with it, what decisions would you make? So that's going to be the one question that's probably going to take you 3 to 5 minutes on. We have a total of 48 questions, of which the total points are 50 points, pretty much equally distributed between Dr. Hogan and I as far as the material content. And then there's one extra credit question in there, which you should be able to get, and that'll go directly to your total score. And the total score obviously is out of 100, maximum of 101. And we've had that in the past. Students have actually aced it and got a 100 plus one question right. So we'll see how you've done. I will say before I go to my questions, I want to compliment you all on your research reports, especially. There were some really outstanding, well-written reports out there. And that's why I really want to encourage you guys to download them. Great references. Yes, we had a lot of similar topics. Osteosarcoma was really popular and hemangiosarcoma was really popular. But we had a very broad range of really well-done reports. And you guys know who you are because I wrote you comments about those. And I think it's really important to, as veterinarians and student doctors, as I like to call you guys, we have a lot of different aspects that would define our ability to be good veterinarians. One is obviously having the knowledge, which you're working toward. Two is being able to communicate that knowledge. Writing is very important as well as oral communication. Hopefully you're getting a feel for that. But also, you learn to write better when you work together. And that was a good lesson. I'm sure Dr. Hogan can share his experiences when he was doing his PhD and working on his first writings. When I was sitting sitting at the desk that you guys are now at, obviously not sitting at our desks over in the lecture hall, I was pretty intimidated as a writer. I recognized I was not a strong writer. And it was through collaborations and working with, and I think I told you the story, my very first paper went through 7 revisions before I went out, but it was a techniques paper, and it's one of the most cited papers I have in my repertoire of published articles. I'm really proud of that. It was because I was taught how to write well, and it was something I had to work at. And now with humble confidence, I can say I've achieved that. And I want you to appreciate that. But a lot of times you get better writing when you work together. And that was one of the lessons of this whole project, as well as seeing how cell biology is so pivotal in how we function in medicine. That's another important aspect. And cancer being one of the most common, well-studied areas that we still need to work on, because this is a disease, broad disease, that many of you identified, that affects our animal population, our pets, as well as us. So it's very relevant across both the human and animal bond story. So once again, congratulations, and please do download those. Those are for you. This is the first time I've done this. It's uploaded. It took a little bit of time because I had to remove all your names and scores. Obviously, you guys now know why I asked for the doc file. But there's some really well-written ones in there. And I really think you all would benefit from reading it, plus having it as a reference, as I said, for oncology. It'll impress the heck out of the oncologists when you have a good foundation on a lot of these tumors. And then also in your clinics and on your boards. So that said, my style of questions, as you already knew, one of the reasons Dr. Hogan and I gave you those quizzes is kind of get a feel for our style of questions. He is, as you know, is an outstanding virologist, and he's very mechanistically focused in this course, and that's his drive for it. I'm a little bit more blended as a clinical researcher in the sense that I, have a different focus, and that is what is the link between clinical medicine? What the idea being is how can we help you learn something and retain it? Can you link it to a disease? So that said, a lot of my questions are going to be focused on the clinical aspects of the topics that we talked about. I do expect you to have a good foundation on the topics and be able to explain them. So some of the multiple choice questions will reflect that. But a lot of the questions I have will have a clinical spin. Nothing overwhelmingly demanding this stage, because I know your limitations and your knowledge base, but I wanted to ask it in such a way that you can apply it. So this gives you a sort of a foundation, okay? So this is a post-translation modification question. I kept it open so you can see all my questions, but also so I can actually hopefully also manage the chat. So I also teach in Venn. I don't know if you guys are familiar with that. Part of what we do with Venn is we also have question and answer sessions, and I can always see the answers, but I'm trying to figure out how this is. One thing about Zoom, which is weird, you can't pull up. Can you pull up the chat section?
We'll let you know if any questions come through the chat, Doc.
Well, what I'm curious about is what I'd like to do is give you an opportunity to chat. So here's the first question. Blank is the addition of carbohydrate group to protein and primarily occurs in the blank. I was wanting to use the chat so you guys can basically provide the answers.
Oh, it just popped up. Let's see if I can see it. So, who wants to take a stab at it? Unlike Dr. Hogan, who was very kind to give you the answers, I'm not going to give you the answers. I want you guys to give me the answers. So, who wants to take a stab at it? Chat.
It's glycosylation, and it primarily occurs in the Golgi body.
Okay, is it? Glycosylation is correct.
I thought it was in the ER because the endoplasmic reticulum is for modification.
Very good. The ER is the primary location. So the answer to this one would be glycosylation and ER. So if you had a question like this, half point would be to glycosylation, the other half would be ER. Very good, Gina, and very good aim as well. Good job. So guys, feel free to post your answers in chat when we do the next one. All right, so this is another fill in the blank. So you can see with this one, I'll have to go in and if I were to give this question, because there's plenty of different globular proteins and various fibrous proteins, but what comes to mind as a clinician to be, what's a good example of a globular protein? One very relevant to what we're dealing with right now. What would be your answers? Kick it in there, guys. Feel free to throw it in Zoom in the chat.
Immunoglobulin.
Okay, antibodies would be one. Very good. How about fibrous protein? There's plenty out there. Think about anatomy. Think about what we talked about when we were doing palpation of the dog and we were examining different tissues.
Collagen.
Very good. Collagen and fibrin, particularly, you could also say tendons and ligaments, and that would be correct, right? They would be composed in that. So those would be the questions I would be looking for. In this question, it's a little broader, so you could give me a number of answers and they would be correct. All right. How about this one? Ion channels are composed of membrane proteins which facilitate the passage of hence ion channels.
Calcium.
Calcium. Very good. All right. That one, to me, I thought was pretty straightforward. Well, here's a clinical one to spin a little bit. We talked about this in mitosis, and there's also different, and I would just, and I'll emphasize this in just a minute, but if I'm giving you some background information about karyotype, I would also expect you to know some of the diseases in which this karyotype is shifted. So what's the answer to this one? So do you remember what, this one should be pretty obvious. What we all study this in biology. What species is this?
Humans.
That's right, very good. What species is this?
Dogs.
Yeah, very good.
Cat.
There it is. Very good. Excellent. All right, another multiple choice. So apoptosis, as you remember, we talked about apoptosis, is a normal physiological process mediated by either intrinsic or extrinsic signals. What's the difference between the two? Do you remember?
Intrinsic comes from within the cell itself, causing its own cell death, and then extrinsic is from some external force.
Okay, excellent. So BC2, it should be BCL2 and BAX, or BC2. What would that be, an intrinsic or extrinsic signal? Ah, very good, Elena. You guys are giving the answer, but that's very good. Intrinsic or extrinsic?
It's intrinsic, the first one.
Thank you. How about B?
Also intrinsic.
Very good. How about C?
Extrinsic.
Very good. And D?
Extrinsic.
So the answer would be
E.
very good. So that's the flavor of questions that I ask. So mitotic index is a good question. Mitotic index probably would be a good question on the final exam. Hint, hint. And I'll be honest with you, that I call it my thinking question. My case, I give you a nice factual case that be highly relevant in the clinics, and I ask some very important questions. I give you information in that case. Read the information that's in there. Think about what's provided, and then you need to be very broad in your thought process as how you would handle that case. Okay. And you have the necessary knowledge at this stage to make some decent decisions. And the nice thing about it, and when it's written, is Dr. Gogol can read it. And if I find that I'm happy with the majority of your answer, you get full credit. All right. I have been surprised and impressed many times when students provide me things that I've never considered that are equally acceptable as a treatment choice. Hint, hint. All right. Any questions, guys? Please feel free to ask us.
Dr. Gogol, how many questions or how many points is the test supposed to be?
The point system is 50. So you should know now with two 10-point quizzes and the 30-point report that you have upwards of maximum 50 points going into the final. And if you look at our syllabus, we are in line with what the grade scale is relative to the point system. For the final, you have 50 points plus the 1 extra credit point. That's my gift. So you got like a bonus point, which I am reasonably confident that you will be able to answer. So that one will be added to your point. So you know the point structure, and it's just additive. So if you get 101 points, you still get an A. Get my drift. If you were to get 48 points on the exam from our 50 questions, you get the 1 credit point. Now you got 49. And you add that to whatever your current point system is. If you've got a 48 total going into the final plus 48 or 49, you kind of know what your score would be, like a 97. Does that help?
Yes, thank you.
You're welcome. Dr. Hogan, you still there, sir?
Yes, I am.
There's a question from Adelaide. She says, I have a question. Dr. Hogan talked about glycosylation in his lecture and emphasized the Golgi body. Do more proteins get glycosylated in the ER than in Golgi? That's a good question.
Yeah, it's a good question. Usually it's finishing that's done in the Golgi. So you get started with the process in the ER, and you have a lot more processes than just glycosylation, but it gets started there, and then and we don't go into a whole lot of detail on it because glycomics is a whole thing. And I'm not very good at it, to be honest with you, but no, just enough to be dangerous. But it gets started there, then it gets shipped over, and that's when the branches are added to it, right? So you get a lot more decoration of think of it as a Christmas tree, right? So you get your Christmas tree, put it down, but then you got to add everything on top of it, right? So it's kind of the same process. Hopefully that gives you a good kind of visual and can help you understand it a little bit better.
So technically both answers would be correct. It starts in the ER. I guess I could have reworded that question a little bit differently. It's interesting.
Well, and again, it's not, and I'm sure you could find an example where everything was done in the ER, and then you can find for sure examples where it's got to go through finishing in the Golgi, right? Yeah. So I think in biology, you look hard enough, you're going to find whatever you're looking for.
It's not black and white, is what you're saying.
Yeah,
I agree.
Yeah.
Any other questions? Well, on behalf of Dr. Hogan and myself, we really enjoyed working with you all and getting to know some of you more than others because of the way the course is laid out. We are somewhat reticent in the sense that this course usually starts at the beginning of the semester, and we're usually done in September. But hopefully, maybe as a reverse way it's being taught this year, you can link some of the things you've learned in other courses to a better appreciation of how important understanding the biology of the cell is in disease processes. And I truly do hope that I think a lot of you did enjoy the report and were able to see how we made a lot of the connections or what our topics that we discussed fit into how we understand cancer and what the new directions or the current and the new directions are for dealing with cancer. I mean, as a summary, I mean, Dr. Hogan, you're welcome to kick in as well. For someone who does a lot of cancer biology work, I think our goal obviously is looking at mechanisms in which we can assault the cancer from multiple directions. And I think you all can appreciate that once tumor formation occurs within the body and the host and differentiation occurs, you're not dealing with one particular tumor cell, but a very heterogeneous population, which makes it very challenging for treatment. So it points, really part of the points the finger. I answered some of your classmates went in certain directions, and I kind of commented in some of the reports, which you all didn't get to see, but those individual groups did, that really what we need to be looking at in addition to different therapies is finding better ways to diagnose neoplasia and cancer earlier. And maybe even look at genomics to see when individuals, animals, and people are more susceptible to different cancers, because we know there's a certain hereditary component. But we're all faced by our development, our functionality, our existence is foundation of two things. One is the genetics we inherit from our parents, and the other is the environment that we are exposed to as we develop and grow. And that environment starts in utero, goes throughout the rest of our life. And the same story goes with animals. So those are the things that we need to be aware of as clinicians and as researchers. And those are the things that we need to focus on. Last minute question, how much time are we allotted for this exam? We've been very generous, Julie, so you should have 2 hours for it. My experience, Dr. Hogan's and my experience is that I would say a good percentage of class is done within the first hour. You always have that gifted individual that can walk out in 28 minutes, and we just kind of just shake our heads and go, wow, because I was never like that. I don't know about you, Dr. Hogan, but I would encourage you to take your time. There is no, there is no extra credit for finishing early. I do, the strategy, as Dr. Hogan talked about multiple choice. Each answer on multiple choice is true, false. That's why I always did it. And then I would come back, and then usually I did pretty well. And your first impression is usually the correct one.
Yes. I'm not to interrupt you, but I'm glad you brought that up. Yeah, balance your time. I think, and you are absolutely welcome to use the full 2 hours. But I think if you get to that point, you're overthinking the questions, and you're probably doing yourself more harm than good. And it's not going to affect us this year where, we usually would tag team where, Bob would take the first hour, I would take the second hour. It's fine. It's totally fine. But again, don't overthink it. If you're there at 90 minutes and you've got 4 questions that you just can't, figure out, what you probably either A, don't know the material well enough, and that's not a dig on anyone. That's just a section that you probably didn't study as much as you probably should have. And that happens to all of us. But don't overthink it. Like I said, look at each one of the answers and decide on its own, is that correct or is that not correct? And this is where paper tests are actually great, because you can literally scratch through the ones that are correct and aren't, and narrow it down. And that will help you tremendously.
You're right. That's what I used to do all the time.
Yeah, it's a process
Sometimes
of
it
elimination.
came down to 2 answers, and I go, and then have to go back. Well, sometimes I would encourage you also to think about this. I'll read a question, and sometimes I misinterpret what the question is. So make sure you take your time reading the question or the statement carefully, because we're all guilty of sometimes inflecting our own interpretation, our own wording into it, and realize, whoa, I didn't read that right, or I interpreted it wrong, and I chose the wrong question because I chose the wrong answer because I read the wrong question wrong, is what I'm trying to say as well. So
those other things we can tell you. So I did record this. I'm so proud. And